All right. So, in this lecture, let's talk about how sequence design priorities change based on the application. We already kind of know that uh, no universal best mRNA sequence exists. It completely depends on what you are trying to do clinically. Are we trying to replace a missing protein? Do you want to trigger an immune response? Or are you editing a gene? So each of these needs a different behavior from the RNA. And it's not just mechanism, expression kinetics also matter, right? Some therapies want a big spike and then rapid clearance. Others need moderate but sustained production over days or weeks. So then again, we have this immunogenicity. So for vaccines, immune activation is the goal, but for protein replacement, not so much. And practically speaking, right? So dose and route of injection also change everything. Manufacturing limits what you can actually make at the scale. And every therapy has a safety window before toxicity shows up. So, based on all these things, what you need to understand is that you are doing a constrained optimization under, clinical requirements. Now let's talk a little bit more on those applications. Most therapeutic mRNA programs fall into three buckets: vaccines, protein replacement, and gene editing. And each one wants something different. Vaccines are designed to trigger the immune system, so you want a strong antigen production, but only briefly. So once immunity is primed, continued expression is unnecessary and just adds inflammation. So that means we need high peak and short duration. Now, when it comes to protein replacement, that's basically opposite thing. You are restoring a missing function, so you need a stable protein levels over time. I mean, too little, no effect, right? And too much, not good. That may lead to toxicity. So the goal is sustained, predictable expression. Gene editing is again different. So the editor only needs to be around enough to modify the genome. After that, continued expression may increase off target risk. So you want tightly time limited expression and then rapid clearance. So what we just saw is that for the same molecule, we want different temporal requirements based on what needs to be achieved. When it comes to sequence priorities for vaccines, the main goal is antigen production. So, what matters most is getting a strong burst of protein early after delivery. A high codon adaptation is important to minimize elongation rate. Maximize elongation rate. A minimal structure in the 5' UTR is helpful, So ribosomes can initiate efficiently. Shorter coding sequence can even help because you get more protein per ribosome cycle. And here, stability is not the main concern. You want enough expression to prime immunity. And then immunogenicity constraints are also, different here. Some innate immune sensing is acceptable and actually even helpful because it boosts immune activation. If we take the successful platforms as an example, probably multiple factors likely contributed, such as sequence design, dose, immunogenicity, but more importantly, this modified nucleotides obviously improved translation efficiency and tolerability. Now, when it comes to large scale manufacturing, the constraints shift again. Some things you have to control tightly. Double strand RNA, for example, you reduce it through purification, but how much forms in the first place also depends on sequence design and transcription conditions. You also need consistency at the 3' end and reliable cap incorporation. But what's interesting here is that vaccines are tolerant, in other areas, things like side effects such as soreness, fever, fatigue. So these are temporary and again expected. So and moderate variability in a poly length is usually fine. So overall, the idea is not uh, perfect uniformity everywhere. It is selective control. So you need to tighten what affects safety and dose consistency. But now, when it comes to protein replacement, it is a different problem from vaccines. So here, you are not trying to spike expression and let it disappear. You are trying to recreate a missing physiological function, for example, and sometimes for weeks. So that means expression has to be stable, predictable as well. Immune activation becomes a major problem here. If the innate system shuts down translation or prevents repeat dosing, so the therapy is no use. Efficiency still matters, of course, but not for speed. It matters because you need sustained protein levels at tolerable doses. And toxicity tolerance is different. Patients may receive repeated dosing over long periods. So safety, margins have to be tight. And again, manufacturing consistency obviously becomes critical. So the design objective, shifts a bit. Vaccines optimize for peak output, whereas protein replacement optimizes for durability. And when it comes to sequence design for protein replacement therapies, it is driven by basically durability. You want RNA that persists, translates efficiently over time, and doesn't trigger innate shutdown. So that means engineering the 3' UTR for stability, not just making it longer, but embedding regulatory elements that slow degradation and support the right protein organization. Poly length becomes, kind of a stability control knob. Most therapeutic designs push around 100 nucleotides or more. Structure is another tuning variable. You want enough to protect the molecule, but not so much that the ribosomes stall. So stability is, engineered more regionally, not globally, really. Now, immune silence becomes a hard constraint. Unlike vaccines, innate activation is not helpful here. It directly reduces expression and limits repeat dosing. So, uridine content gets managed. Some of that is sequence level, that is, removing U rich motifs. And then some is actually chemical, that is replacing uridine with modified bases like methyl pseudouridine. And double strand RNA control is critical. That starts with sequence design to prevent formation, but also requires manufacturing purification. So, overall, the design philosophy flips compared to vaccines. So, speed is no longer the goal. Longevity and immune invisibility are important here. So, when it comes to engineering protein replacement therapies, there are many things you need to pay attention. Uh, the 3' UTR acts like a regulatory control panel. It has bunch of binding sites that either stabilize the transcript or recruit degradation machinery. A poly tail length is important also. Longer and more uniform tails generally slow deadenylation and extend functional lifetime. Then there are specific sequence motifs. Commonly these AU-rich elements that actively recruit decay pathways. So if you remove those, you can extend persistence. Structure is another major important point. You want structure that protects the RNA, but not so much that the ribosome cannot even move efficiently. And there's a translation itself feeds back into stability. If ribosomes stall, due to local structure or codon issues, the cell interprets that as a defective transcript and activates quality control pathways. So the point here is that persistence is not a single parameter. It is an emergent property of structure, motifs, and ribosome dynamics. From a modeling perspective, this is where structural energy predictions and regulatory motif counts become useful features. Let's talk about protein replacement therapies, manufacturing constraints. Um, so one thing is that these therapies may be given for months to years. So that changes the risk model completely. First of all, this double-stranded RNA tolerance is lower. Even small amounts of innate activation becomes a problem if the patient is exposed repeatedly. And then the 3' end must be tightly controlled. A variability there directly affects RNA lifetime, and that means unpredictable drug exposure. If patients are dosed over and over, even small fractions of abnormal species can accumulate. And finally, manufacturing consistency becomes critical because each dose must behave the same as the last one. Otherwise, pharmacokinetics drift over time. And now let's talk about gene editing applications. So gene editing is obviously different from protein replacement or vaccine therapies because the a biological change is permanent here. You only need the editor long enough to modify the genome. So you want controlled, limited expression, just enough to achieve editing, but not enough to cause risk. So timing also is critical. The editor must be present briefly, then cleared quickly. Persistent nuclease activity increases off-target damage. Innate immune activation must also be minimized. So here, the entire design idea is transient exposure with maximal precision. So when it comes to a sequence priorities for editing, so the most important point is intracellular editor exposure. How much is present and for how long? Editing efficiency saturates relatively quickly, but off-target risk keeps increasing as exposure accumulates. So you are not optimizing for maximum protein production. You are optimizing for controlled, limited presence. That means you typically avoid strong translation. So moderate expression is enough to achieve editing. And you also want transient functional lifetime. Once editing has occurred, continued editor activity provides no benefit. And structural features of the RNA are used to tune translation timing and degradation kinetics. So the editor is present just long enough to do the edit, then gets cleared. So overall, editing design is fundamentally about exposure control, not maximum expression. Now, let's talk about delivery context. Different tissues have different immune sensitivities. A muscle and lymph nodes, for example, they can tolerate innate activation pretty well. Actually, that's one reason why vaccines work well with intramuscular injection. But the liver is different. When you deliver, IV, LNPs accumulate heavily in the liver. Kupffer cells and hepatocytes detect the RNA through RIG1 and TLR pathways. And here is the problem. So when these cells activate, they release cytokines directly into circulation. So because the liver is so vascularized, this can rapidly produce a systemic inflammatory response, fever, cytokine release, even hepatotoxicity. So the difference is not whether activation occurs. Every tissue can detect RNA, right? So the difference is whether activation stays local or becomes systemic. Route, dose, biodistribution, organ-specific sensing, repeat dosing feasibility, local RNA's environment, all of these things changes the design constraints. Okay, so what's the reality? Sequence design is not a single optimization problem. If we want longer persistence, we generally uh, add stabilizing structure, but structure slows ribosome movement and can reduce translation efficiency. Maybe we want a translation bit too fast, but proteins may not fold properly during synthesis. And we know that poly A tails improve stability, but longer tails become harder to manufacture consistently. And double strand RNA is the classic process trade off. The lower you want it, the tighter your upstream design and downstream purification have to be, and that drives up cost. So there is no universal optimum. Each therapeutic application chooses a different balance depending on what matters most clinically. So when you are designing a therapeutic RNA, you know that you are not searching an unconstrained space. So some constraints define what's even physically or biologically possible. So these are hard constraints, things you cannot change basically. The protein sequence, must remain unchanged. The transcription template must be valid. Translation has to initiate properly. You cannot change these. So everything else is optimization. You shape RNA structure, codon usage, GC content, immune visibility to improve performance. But uh, these are, tunable objectives, not absolute requirements. In modeling terms, hard constraints define, feasible set space. Soft constraints define what counts as optimal inside that space. So, end of the day, there isn't one objective function for best mRNA. The weights change with the therapeutic job. So vaccines want a high peak and don't care about a long half-life. They can tolerate more innate sensing. So protein replacement is the opposite. You want a predictable, durable expression and very low innate activation. Gene editing is the strictest. You need enough editor to cross an activity threshold, but only briefly. So, duration control and off-target risk dominate the loss function. So, same design variables, completely different weights depending on the application.